Thanks, Mark. Can everyone hear me okay?
You can pull it out.
All right. This is a little talk show.
Maybe.
Oh, okay, gotcha. I'll stand up here. So I want to start off with a picture of some bacteria and fungi in the built environment. These are some scanning electron micrographs that a postdoc in my lab put together. This one here in the middle, the blue one, this is a biofilm on a cutting board. These other two are actually door trim samples, so dust samples inside the home. And I want to start with this to point out that we're dealing with organisms here, not DNA sequences. And I know I'm preaching to convert and so forth, but I mention this because for a lot of these organisms that we find in the built environment, we do actually know quite a bit about their natural history and their ecology. And it's important to keep that in mind because we have these new methods that are available now, but there's a lot of microbiology history that we can pull from to start understanding some of the patterns that we're seeing in the built environment. And I'm standing up here now giving a talk, but I want to emphasize that this is really a large interdisciplinary project, and it's easily one of the most collaborative projects I've ever been involved in, and it's really been a lot of fun. So some of the people are here in this room. So there's folks from my group in the Department of Ecology and Evolutionary Biology at CU here, and Shelley Miller's group in Mechanical Engineering. And if you have specific questions about the built environment stuff, they're the ones that you should ask. And then also, they're not here with us right now, but Rob Dunn and Holly Manager have really been critical to this project. So I want to first start off by a pilot study that was just published yesterday, actually, where we essentially asked the question, what factors influence microbial diversity? So diversity and composition found in the homes. And this is just a pilot study. We sampled 40 homes near Raleigh, North Carolina, and we're sampling 9 standardized environments in each of those homes. So we just wanted to get sort of a broad brush, like what kind of diversity is out there, what sort of environments might be useful for more detailed analyses. So we sampled, door trims, and the idea behind the door trim is very few people clean that environment, so it sort of accumulates dust and so forth. So the idea is we could kind of use that as a proxy for what's blowing around in the air inside the homes, and also the door trim outside the home to see what's out there. And a wide range of other environments, and we picked these because we thought they'd have distinct communities, at least bacterial communities, and also because almost every home had them. Okay, so I don't want to, and this is the general sampling technique. It's swabs, and it's quite easy to do this without contaminating the swabs. You don't even come close. This is my daughter, though, so she's not so good at doing the sampling. Because what you don't see is immediately after the swabbing, she wanted to sample her diaper. So we practiced aseptic technique. She was practicing septic technique. So just some key take-home messages from that work. One, what we found and what lots of other people have found is that skin taxa dominate most indoor locations. Not all, but in most of these locations, we see a lot of skin-associated taxa. And again, not surprising. We've actually really known that for 50 or 60 years at least. But it's useful to point that out. And we also see this, so we see this across locations within homes, and even if we hone in on one environment, the kitchen, this is some work done by my postdoc Gilbert about a year ago, we can see that those locations in the kitchen that are touched more frequently, like the pad on the microwave, the door handles, the handle of the refrigerator, have much higher abundances of these skin-associated taxa, just due to the transference of bacteria from the skin to these surfaces. The other thing we see is there's a lot of differences across sites, so I'll spare you the statistics, but all of those 9 sites that we sampled harbor distinct bacterial communities. But there is a lot of spread. There is a lot of variability within homes, between homes, which of course isn't surprising. But a lot of those differences appear to be driven by differences in source inoculum. So in most of these locations, there's probably not a lot of community turnover, probably not a lot of growth going on. These microbes probably got there from somewhere else. So we can start linking these differences in the bacterial communities on these different locations to different sources. And you can ignore the numbers for now. You ignore the numbers because it's very hard to quantitatively identify indicator taxa, but what matters is the colors here, so it emphasizes the relative importance of different sources on these different locations. So a bunch of the things are very obvious. For example, on your toilet seat, you have a lot of skin taxa and fecal taxa. Not surprising, but thankfully that's not the case on the kitchen counter, where we see a lot of skin taxa as well as quite a few taxa from leaves, and most of these are probably produce-associated taxa. And for example, in the outer door trim, we see a lot of taxa from leaf surfaces, probably getting whisked up off of trees and grasses and so forth outside, and also a lot of soil or dust-associated taxa. The other finding, so we see a lot of variability within homes, and the other thing that was interesting is we're starting to see some major patterns emerge when we look between homes. So what's explaining the differences at a given sampling location across the 40 homes that were sampled. And one thing that immediately jumped out was this huge dog effect. And this goes back to what Jordan was mentioning just a bit earlier, but in this case we're looking at bacteria, and that on almost all the environments that we sampled, except thankfully the kitchen environments, most people have fairly obedient dogs that are jumping up on the kitchen counter and so forth, we see a strong dog effect. So in other words, homes without dogs have very distinct bacterial communities compared to those with dogs. And you can actually trace this back to specific bacteria associated with dogs. So it's not that they're just tracking in dirt or something like that. They actually have taxa are far more abundant on their fur and their mouths, and they're leaving those bacteria on pillowcases and other environments inside the home. But one thing I want to point out is this is really just sort of an initial, again, this is a pilot study. We're just getting at the low-hanging fruit. What we really want to know is, what are the factors influencing microbial diversity found in homes, both the biotic and the abiotic factors, and perhaps more importantly, which of these factors are most important in structuring the microbial diversity we found in different locations. So we have a two-pronged approach. One is to sample broadly. So we call this, it used to be the 1,000-home study, then we got a little too energetic, and now it's the 14-home study. So this is 14 homes that have been sampled across the U.S., and this is carried out by citizen scientists, in other words, people that are not only helping us get the samples, but they're actually engaged in the scientific process. And then a subset of those, only 15 of the homes located nearby for logistical reasons, we're sampling much more intensively and getting a lot more data on the conditions within the homes. So I just want to briefly talk about the 14 Homes project. So this is a map of the 14 homes that have already been sampled. So in other words, these individuals receive a kit to do the sampling, very detailed instructions on how to do the sampling, and perhaps most importantly, they have to fill out an enormous questionnaire. That took the most time, was to figure out all the questions we could possibly ask these people to get information on their home, what they're doing in their home, who lives in the home, what attributes their homes have, and so forth. The advantage of this sort of sampling strategy is we have homes that vary from log cabins in the middle of the wilderness to apartments or townhomes in cities and so forth. So we have a broad range of different types of homes, different types of occupancy patterns, and of course different locations. I want to point out that the homes in orange here, these are participants in the Personal Genome Project. So for those homes, it's about 400 of them, we have very detailed medical records available for at least one of the occupants living in those homes, one of the occupants living in those homes. So we have every state, both urban and rural, and I believe it's at least 6 homes per state. And they're sampling, in each of these cases, they're sampling 4 standardized environments: door trim outside the home, door trim inside the home, the kitchen counter, and the pillowcase. Again, this is largely selected because we already knew these were very distinct communities in these different environments. And again, we're using this door trim as a proxy for the types of microbes we're likely to be seeing aerosolized inside the home. And this is what one small subset of these 5,600 samples look like. These are now all in my lab about a mile from here, and I think if you listen carefully, you can hear the screams of my technicians. Now they're trying to process these samples. Yeah, so enormous amount of samples, and this is really, I really have to give credit to Rob Dunn, the Holly manager. They pulled all of this off in about 4 months, from recruiting volunteers to actually getting the samples back, or maybe about 5 months. But they did it very rapidly. And the participation was very good. People took the time to fill out the questionnaire because they had to, otherwise they wouldn't get the kit. So what we're going to be doing is direct PCR amplification to process these samples, and we're going to be looking both at bacteria and at fungi, and both of these will be on the Illumina sequencing platform. And for fungi, we've developed we, among others, have developed a protocol to target the ITS1 region. So it's a short region, so it's amenable to sequencing on the Illumina platform, but it gives a fairly high degree of taxonomic resolution of the fungi. And we've used this for about 100 air samples up to now and about 600 soil samples, and this is just some example from soil samples. And I'm just showing sort of broad taxonomic groups of fungi. But this is really a useful strategy to not only, we can get the bacterial diversity, now we can start looking at the fungal diversity across these 1,400 homes. So the idea is we can start using these samples to look at questions about how location of the home structures the microbial diversity found within homes, how lifestyle or the number of occupants or what type of occupants you have there, how materials used to build a home, the operation of your HVAC system, all these sorts of questions that we can get at by having 1,400 homes sampled across. We couldn't do this if we only had 30 homes or 40 homes. It's impossible to address some of these questions. And hopefully we'll be able to get at some of the questions that Jordan alluded to, looking at links between asthma and potential microbial organisms living within the home. And so the scientific value remains to be determined, so we don't have the data yet, but arguably, we already know that it's had an influence on outreach efforts and sort of getting the public engaged in this process, because each of these 1,400 homes, they've not only received the sampling kits and filled out the questionnaire, they've also received a lot of information about the project, why we're doing the project, and both Holly and Rob at NC State have spent a lot of time thinking about how to give the data back to the people, not just give them bar graphs or ordination plots, that would be horrible, but actually give them the data back in a useful manner so they can interpret the results from their home and think about it, and also engage in the process of generating hypotheses about the data. So I think that's really already been an interesting part of this project, and the feedback has been really great. So the problems with this project, of course, is whenever we sacrifice sort of breadth, we sacrifice sort of accuracy or detailed investigations of individual homes for capturing 1,400 homes all across the U.S. So we don't have direct assessment of environmental conditions or air quality metrics. We tried to ask as many questions as we could about the composition of the home and HVAC condition and the conditions within the home, but of course, for many homes, it's difficult to get that information. And it's also only a single time point sampling. There's no direct air sampling in this study, and there's no quantitative measures of cell abundances. Okay, so this is where the second component of this project comes in, where we targeted 15 of these homes, a subset of the larger homes, for more detailed investigations, where we could start filling in some of those gaps, and also looking at temporal variability within the homes. So this is 15 homes that are sampled twice per season, and this is both the swabs, the same swabs that are collected for the 14-home project, as well as actual direct indoor and outdoor air samples. It's 24-hour samples. We finished all the winter sampling. I'll show you some results from that. The spring sampling is about to finish up fairly soon, in a week or so. And the same type of analyses, we'll be looking at both fungal and bacterial communities by amplicon sequencing. And what's unique is we'll be looking at cell abundances via flow cytometry, at least on the air samples, to get measures of the numbers of cells that we're seeing in these homes. And so this is definitely where, so there's a long list of analyses that are being done on these homes to characterize the home environment. So walk-through home inspection and blower door tests, measures of carbon monoxide, CO2, relative humidity, temperature, both indoor and outdoor at each sampling point, ozone, VOCs, PM10, PM2.5, both the concentrations and some measures of some characterization of the aerosols, and assessment of ventilation potential. And I think I'm missing some here, but I want to point out that John, who's been very involved in this project and engineered this whole system, he has a poster that will be right after this. So if you have more questions about what sort of measurements we're taking, you should definitely ask him. But the one thing I want to point out, so one issue that we quickly came up with, that quickly arose when we were doing the sampling, is that these pumps are loud. In the first 5 minutes, you're like, oh, no problem, I can live without for 24 hours, and then like 4 hours into it, you're like, it's drilling into my head. So I have to thank John and Pat, who engineered this marvelous, so the pumps are in here, they're in this little cooler that provides some sort of soundproof, and then they built this muffler system to really, and it works wonders, and now my wife won't tell me when they're sampling my home, because it really has dramatically reduced the volume of the pumps. And so just to give you a feel for some of the data, so this was really cool to me. I know this is old hat for the building scientists here. So on the bottom here we have the activity diary, so what people are doing in their homes, when it's occupied, where they are in the homes while they're there. And on the top we have some of a handful of the measurements that are being taken. And just a few things I want to point out is, one, if you look at the blue line here, this is the PM10 concentration, so aerosol particles, 10 microns or less, over the 24-hour period, just from one home to show the patterns here. But what's pretty cool is you can see every time they start cooking, PM10 concentrations go up, like within minutes, which is pretty neat. And the other thing that's neat is you can see at night when the heater is on, this is forced air heating, you see the PM10 go up every time the heater goes on. You can see that both through the fluctuations of the temperature and the PM10. And then of course also if you look at CO2, they opened up windows. As soon as they open up the windows, the CO2 drops. So it's pretty cool to me, because just from looking at this data, you learn a lot about what people are doing in their homes. It's almost creepy when you find out about you know exactly when they woke up in the morning, for example, because you see that spike in CO2. You know when they started their coffee, and so forth. And then of course we're also sampling outdoor. By the way, this is the microbial air filter that's in there. So we have both paired indoor and outdoor sampling occurring at each of these homes. So just a glimpse at some of the results. We just got the data back about a week and a half ago, so this is just, and again, this is only the first season of sampling. This is just the winter sampling, so we only have 2 time points from each of the 15 homes, and the project will end in the fall, so we still have quite a bit more sampling to do. So one thing that's interesting is if we look at the temporal variability within homes, so if we compare the 2 time points that we already have, some sites are a lot more variable than others. And it's not particularly surprising. So the less variable sites, for example, are the pillow and the floor. So pillow, basically, in almost all homes, it basically looks like slobber and skin, more or less. And so some of these environments are quite stable over time. If you were to go back and resample, and in all cases, these are sampled at least 3 weeks apart, the communities look fairly similar, okay? But other communities or other environments harbor bacterial communities that are turning over much more rapidly. They're changing much more dramatically in composition between 2 different time points. So for example, if we look at the indoor air or the outdoor air bacterial communities, they're quite distinct every time we sample. And what will be interesting to see is it can start, of course, predicting why you get one type of community versus another and so forth. So this is perhaps not surprising, but it does show that that a single time point sampling of indoor air may not necessarily be that informative, right? Because it's only at that time that you sampled, and the next time you sample may be completely different types of bacteria found within the home. The other thing that was pretty neat that emerged from this is we also added and collected HVAC filter samples. So this is John crawling into somebody's basement to swab their HVAC filter. And the filters were actually replaced every season, so we can start with a clean substrate every time we go in. What was interesting, and I'll spare you the statistics, but there's no significant difference between the HVAC filter bacterial communities and those found in the actual indoor air samples. And so that's pretty cool, because what it suggests is you can start using the HVAC as a way to sort of passively sample or more readily sample the air inside homes, or at least get integrated samples of bacterial communities found in indoor air over time. And you can see this just at a very coarse level, the phylum level of resolution. And to admit my ignorance here, so at first I was really surprised by this. I was like, no way, the HVAC filter samples should look like outdoor air. Because of course, I had always assumed that the intake for the HVAC filter was outside, not knowing anything about building engineering. And then, of course, I mentioned it to Shelley. She's like, no, the intake is inside the home. So it makes sense why the indoor air community would look a lot like those that are found on the filter sample. So that's just a crude example of some of the utility of working with building scientists on this. Because some things that are very obvious to building scientists are not at all obvious to biologists. Some other results have been interesting. Again, this is just preliminary because we only have the first season worth of data. We're still sampling right now. Is this fairly good correlation between the number of carpeted rooms in the home and the percent of the bacterial communities that are associated with skin. So we see this both whether we're looking at the indoor air communities, so these are communities sampled directly from the air over that 24-hour period, the inner door trim, or the HVAC filters. We see the same pattern in all of these environments, where we see more carpeted rooms, meaning more skin-associated taxa. And this is probably just due to the fact that carpet's a good reservoir for a lot of bacteria. So every time you shuffle your feet across the carpet, you're aerosolizing these skin bacteria that were lodged there, and leading to this relatively high levels of skin-associated taxa in these homes. So if you don't want skin-associated taxa in your house, remove the carpet. So again, I just want to point out that this is really a group effort, and it's definitely a project that's still underway. So we hope to have some cool results, particularly from the 14-home project that we should have fairly soon, because those samples are being processed right now. So thank you very much.
So we've done some HVAC sampling before. I was curious, is for the indoor air samples, how long were those collected for?
24 hours.
And with what? Was it just a cutoff?
0.2 micron filter.
Total. The microbial filter is just total.
So it's a TSP.
For the air samples inside
Yeah, for
the
the
home?
indoor air samples, it's a TSP, but over 24 hours.
Yes,
like a
yes,
basic
exactly.
filter? Okay.
Yeah.
So that's perhaps why.
There's a picture, and here's that. Yeah, so you're talking about this filter. This is collecting this is an outdoor one, but the same thing. So yes, it's everything greater than 0.2 microns, presumably.
And what's the flow rate? Just curious.
Help me out here.
There we go. I'm waiting.
10 liters per minute.
10 liters per minute, but it was for 24 hours. Okay, thanks.
And I should also point out that launching this project, and many thanks to Pat, this is an incredible ordeal because every time she samples, she has to move all this stuff, make sure all the instruments are calibrated, and so, and then of course go to each home twice, once to set it up and once to take it down. And also she's been giving the people the data from their homes and interpreting them, so they get something from this, they get to see sort of information about the characteristics, air quality characteristics.
No, I'm looking at your table 1 from your +1 paper, and it occurs to me that the TV screen and maybe the interior door trim samples are pretty close to air samples.
Yep, exactly. And I think what's going on is due to this charge on the TV screen, it's basically the aerosol particles.
Well, there's a sort there. It's not dust. It doesn't have very large. It's not going to have much in the way of large particles. It's going to be pretty much what stays suspended. And it's the door trim on the top of the door. It's the header,
Yeah.
right? So,
Yeah, and I didn't mention it, but the air filters also look very similar to the door trim samples. So for the 15 homes that we're sampling, but not that close because if you think about it, the door trim is probably integrating over, I don't know, however long people go between cleaning the top of the door trim and the door, which could
since
be
it was built.
decades. Versus the air sample is a 24-hour.
except my mother's is
So we won't sample her house. Any other questions? All right, everybody's ready.